in lecture, I wanted to continue on with microbial evolution. I think we left off here with the idea that if you study the evolutionary microbial evolutionary people, when they are looking at this, they can look for biosignatures, stromatolites. We talked about stromatolites, how those are also a very nice thing to study. And you can date those if they're fossilized, but there's current stromatolites too that are living and cooking along just fine. We use the word protocell for really ancient cell types. And there's been a lot of debate over the many decades here of, when they're trying to figure out where does cellular life arise. Now the current thought is that in the hydrothermal deep vents is probably where we see it happening in the ocean. Still up for debate, but that's kind of where they're looking currently. I'm thinking about. So this timeline is kind of mind-boggling because it goes all the way from age of Earth, 4.6 billion years ago. And then you come down to where multicellular life first started. And there's some key points on here that they've pointed out. But one of the things I really like about this time scale, the way they're showing this graphic, is that they're really putting it into perspective that this whole timeline, that yellow swath there, all of life on Earth during this huge stretch of time is all microbial. And it's not until fairly recently that you started to see even the multicellular life forms, and then of course the more complex life forms. So looks like they're pointing it out to be roughly 3.5 billion years ago. You can see that stromatolites are showing up here too, right? And then we'll talk more about bacterial hopanoids. That's a chemical that's found in certain types of bacteria that's very resistant to being broken down. And so they can actually pick that up in fossils. And if they pick that up, it's so unique to certain types of bacteria that they are highly suspicious and certain that that is evidence of bacterial life. And then they're showing some pictures that they've been able to capture from some of the fossilized samples that they have. It was kind of hard to stretch your mind around the idea of, 3.5 to 4 billion years ago. Oh, come on, slide. All right, let's do it this way. Here's another image of where they're starting to think about, okay, how did cellular life arise? And of course, this isn't an evolution class, so we're not going to get into huge detail, but I don't know if you can make it up from where you're sitting. That's a hydrothermal vent. It's called a black smoker. They're very impressive when you see video footage. So these are deep in the ocean, bottom of the ocean, deep down. And so this is a rendition of that. And they're trying to show that you have a lot of hot water coming through these so-called vents and chimneys. They actually refer to them as chimneys. And you can see that there's a lot of different chemicals floating around here. And in this image, they're trying to show the progression of first, you just had a bunch of chemicals floating around, and then you start to get cellular organization to the point where you have RNA. They actually think, have you been learning that in evolution, that they say RNA was probably the first information storage molecule and simpler. And then it shifted into DNA as the stable, more stable. And then you see LUCA. What is LUCA? Remember LUCA from, if you've taken evolution?
Last universal common.
You got it. Last universal common ancestor. And then diversification. And then dispersal. All right. And then you see here, early bacteria, early archaea. So those are two different domains of life that we don't belong to. That's all microbial. All right. And then we belong to domain eukarya. And some of that's microbial and some of it's not. Okay, so there's this idea that you needed, all of these chemicals floating around in a specific spot where they could all come together And then, of course, you need a phospholipid bilayer membrane to surround the right mixture of chemicals to the point where you can say that's a protocell. And so they think that you have this idea of, there's been a lot of talk about lightning helping to create more complex chemicals. And so they're trying to show that here, where you just start out with certain types of chemicals. You have lightning in the vents. It would be that really hot water coming through where you have that exceptional heat. And through this process, they often talk about clay surfaces, which these hydrothermal vents would have. The formations there, they have a lot of clay, and they talk about how minerals can attach to that. And you can get, with heat, you can get more complex molecules. There's been a lot of interesting thought about, you have to get a cellular life form. So that means you have to have a phospholipid bilayer membrane enclosing the right mixture of molecules. This is just a picture of lipid vesicles. So this is a phospholipid bilayer. If you dump lipids into a beaker of water, they're going to form a phospholipid bilayer membrane based on their chemistry, and parts of them that like water and parts of them that don't like water. You'll get that phospholipid bilayer construction as it starts to interact with the water. And so here you can see that they've done that, and they can show that as the phospholipid bilayer membrane is forming, it captured some RNA inside of it. That RNA was floating around in that beaker. And so you can imagine that happening in those hydrothermal vents, perhaps, where you have the right mixtures. I would imagine this would have taken billions or trillions of tries before you finally got something that resembled a protocell. This was a paper that was just published in 2024 where they're looking at the fatty acids, and they're saying, in a pretty simple situation, they were able to get phospholipid bilayers forming. And so, this is just done in a chemistry lab with the right combination of minerals and hydrogen and bicarbonate, and they were able to get fatty acids up to 18 carbons in length. And so they're, experimentally, they're showing that this is doable under the right conditions. And then this next paper was published back in 2018, but this is a really interesting idea where they're saying that DNA can actually stick around in the environment due to these mineral surfaces. And so DNA likes to attach to those surfaces and can persist as just DNA out, on these mineral surfaces over time. And they're saying that that could be a really important way in which we see the evolution of microbial life, where we're going to learn in this class in short order here, I don't know, about probably next week maybe, how microbes are just really good at sharing DNA. They're very generous with each other, and they like to share DNA with each other. And so they're saying that this could have been another way in which microbes evolved with this DNA hanging out in the environment, and they were able to pick it up and acquire those little sections of DNA and use those stretches, express those genes. And you see the progression of the evolution of cells. And so this is from that same paper where they're trying to show a visual graphic, where here they're showing a white smoker and a black smoker deep in the ocean. And then they're showing how you can get DNA attached to the mineral surfaces. And over time, you can get microbes collecting bits and pieces of this DNA that help with the evolutionary process, where you get more complex cell types, new traits, such as the ability to do photosynthesis. Right, here they show the ceramides hanging out. All right, so the long process we're talking about, they're pushing it, they're saying somewhere between 3.5 to 4 billion years ago was the first protocell. And then over time, we get progression of different cell types popping up. And in large part, at least early on in the studies, they really did focus a lot on stromatolites, even though they showed up a little bit later in the evolutionary process, because most of what you see in a stromatolite, we said they're layer upon layer. So these are showing, this is a fossilized one where they're showing the layers, striation pattern here. And so you get these layers upon layers over time of microbial cells growing in these mats. And a lot of them are photosynthetic organisms, which didn't show up right away, because being photosynthetic is pretty fancy. That took a fair bit of evolution for that to occur. So the earliest protocells, they didn't do photosynthesis. But here's a close-up of that image where you can see the striation pattern. So that's just mats over time, with new growth, kind of like tree rings on a tree trunk, that kind of idea where you have the layering. And did you know you can find them here in Ishpeming? So they have stromatolites, Precambrian stromatolites. Those are old. Well, that's 2.2 to 2.4 billion years old. So we have stromatolites even around here. This one has been sampled and cleaned up and polished so it can show a little bit more of the detail. And most of it, like I said, most of it is photosynthetic. And so we call them, in this case, cyanobacteria. That means they're photosynthetic. Does anyone know what cyano means, what that refers to? It's a color, right? Cyan, well, maybe you don't know. Cyano is referring to blue-green. So the blue-green algae idea, please never in my class say blue-green algae, okay? Can we have that deal, please? And tell everyone else out there in the real world, no, there's no such thing as blue-green algae. That's whoever came up with that term should be. It's cyanobacteria, all right? Because blue-green algae are not algae. They're really bacteria, so that the proper name is cyanobacteria, all right? And so those didn't come along in the evolutionary process until a little bit beyond the first cell. So the Chocolate Hills, located southeast of Ishpeming, is where they were able to find these. Aren't they beautiful? So I don't know if they sell any of these around, like at the rock shops around. Has anyone seen anything about stromatolites? And was it the tourist trap or something? Uper's tourist trap? They might have them there. I haven't looked. I haven't been in that store in a while. But anyways, I always thought you had to go to Australia or something like that to see stromatolites. So I was really geeked out when I'm like, what? Ishpeming? That's pretty cool. So, okay, here are some samples of fossils. I take my hat off, so to speak, to people who try to identify microbial fossils. I think that is incredibly difficult work. Very, very patient. But here are some fossils where they were isolating microbes. And then they're trying to show that they're related to current day living microbes. They look very similar. So this idea that, through the process of evolution, we still see these microbial species still showing up. They're still alive and kicking. I mean, look at this sample. This is an electron microscope picture. And in this mess, you got to try to identify a microbe. And as you're going to see in lab, microbes, they're tiny and they all kind of have somewhat similar, there's not a ton of different shapes of microbes. Here you can see some rod-shaped cells here. But if you don't have a really good eye, I mean, that just looks like bits and pieces of rock material. So again, this is something that takes a ton of patience and a lot of training in order to find microbes in these fossils. So roughly, let's stick with 3.5 for now. Let's say 3.5. That's the number I want you to know in terms of when did the protocells first emerge. We think it's, let's say 3.5. Let's be conservative. But just be aware that some people are pushing that closer to 4 billion instead of 3.5 billion. All right, so 3.5 billion years ago, a protocell emerged, and after probably trillions of tries, but of course, that's not all there is to life. You have to have some way to maintain yourself, get enough energy, and hopefully, ultimately reproduce yourself, right? And so this diagram is showing a possible, early start to biochemistry. Like, how could they have run a physiological pathway given there was no oxygen yet, and they hypothesized there's certain gases in the atmosphere, so they knew there was a lot of iron and sulfur, hydrogen gas. They knew all that was around roughly 3.5 billion years ago. No oxygen. So how could they have done some kind of procedure, some kind of physiological pathway? And usually when we're thinking about that, part of that is you need to make ATP, right? You need to get enough ATP to survive and stay functioning. So this diagram is showing a couple of things going on here. You have the microbial cell, the phospholipid bilayer, so that's the cell membrane. And you can see there's a couple of proteins or enzymes embedded in that. And then they're showing up in the box here that if you have ultraviolet light, which of course sunlight is partly ultraviolet light beaming down on Earth. And if you have iron and protons hanging around with ultraviolet light, they will react. And when they react, you'll get hydrogen gas and the ferric form of iron. So Fe3 plus is ferric form of iron. And then the Fe2 plus is ferrous form of iron. Okay. So why do we care about that? Well, what's really nice about that reaction is it generates hydrogen gas. That's a good thing, and I'll explain why. All right, then they're showing this other reaction here. There's also saying if you have the iron sulfide and hydrogen sulfide hanging out next to each other, they can react. And again, you see one of the products is hydrogen gas. So that's a good thing. Why is it a good thing? Because hydrogen gas, if you're a microbe and you have this hydrogenase enzyme, which isn't terribly fancy in the realm of enzymes, you can use that hydrogenase and strip electrons off of hydrogen gas. And that's what they're showing. They're showing 2 electrons coming off. Remember, each hydrogen has an electron and a proton. So you have, if there's 2 hydrogens, you have 2 electrons. So this hydrogenase's job is to strip electrons off of hydrogen. And here you see it's getting, the electrons are getting sent into this pathway inside the cell here, okay? Now, this is a good thing because it facilitates this reaction where you have water and elemental sulfur. And you end up with hydrogen sulfide, which is a gas. You know what that smells like? Rotten eggs. So not pleasant, but, and actually hydrogen sulfide gas is a respiratory poison for us, for microbes, for any life that we know of on Earth. Get too much of that, you're done for. All right. Not only does it smell bad, but it can kill you. So what does the microbe do? The microbe goes, ew, smelly, toxic. I'm going to get rid of that. But that's good because that keeps facilitating this reaction. All right. So if you can imagine this kind of zinging around and around and around, hydrogen gets stripped of electrons, it facilitates this reaction, you get hydrogen sulfide gas, it facilitates this reaction, and just going round and round and round. Well, why would a microbe do that? Well, one other thing to think about, when a microbe is stripping electrons off of hydrogen, what's left over are protons. All right, so if you take an electron off of hydrogen, you're left with a proton, H plus. So now you have to imagine as this reaction is going on and on and on and on, okay? What's happening is you get a lot of protons, protons. I wish the artist had been a little bit more extravagant. They should have put lots of protons. Lots and lots of protons, because then it would be evident that on this side of the membrane, there's lots of protons. Inside the cell, very few protons, or relatively few protons. So now, the microbe, those wily microbes, what they've done is created a proton gradient. Winner, winner, turkey dinner. That's great. A proton gradient is great. Anytime you see a proton gradient across a membrane, whether it's a cell membrane, mitochondrial membrane, chloroplast membrane, that is sweet because now the proton gradient, so you have more protons out than in. Protons will want to balance each other. They can't go through the phospholipid bilayer membrane because they're charged, so that's not going to work. So what do? With primitive ATPase, the microbe has evolved. The protons go through that. And as they flow through the ATPase, it actually, they refer to this ATP synthase as a molecular motor. It actually causes a torque force that allows for ADP and inorganic phosphate to react. And when you have those two reacting, you end up with ATP, the gold star. That's what you want. Okay. So when you look at this, once you dissect it out, and if you're a biochemistry major, I think you'd appreciate quite quickly, this is a pretty simple reaction. There used to be on the third floor a big, huge chart that showed all the physiological reactions in a cell. And if you just stand there and look at it, it's just mind-boggling. All the biochemistry going on in that cell is just stunning. This is pretty simple. All right, so they suspect this could have been, given the conditions of Earth at the time, and where you have these kinds of chemicals pretty abundant, both in the atmosphere and then what you think about on clay surfaces and other things like that, deep in the ocean, that this kind of simple biochemical pathway could work. And in this case, the goal is to create ATP. No oxygen, okay? When we make our ATP, we have to have oxygen. Microbes are like, doesn't matter. I don't need oxygen. All right, so they figured out a different way to do it because there was no oxygen back then. Questions on this?
It's gonna be on the exam.
Yes. So this is a really good example of what protocells were probably doing. Of course, we can't take I wish we could take a time capsule back there and see, but we can't. But the microbial evolutionary people, they say this is probably a likely way in which they evolved a mechanism to get ATP. Yes. Yes, you got it. And so there's plenty of microbes still living today that never touch oxygen to get their ATP. Yep. You're going to learn in this class that microbes do way different things than what we can do. We're like Luddites in comparison to them. They are just amazing. We're pretty simple when it comes to how we have to get our ATP. Okay, so you heard me talk about protons, proton gradient, generation of ATP. Get comfortable with that because in this class, we're going to be thinking about that quite a bit. Okay, so give this some thought and study time. So they really do think that this was could have been a way in which they started piecing together biochemical reactions in order to get ATP, which of course they need that for all kinds of things, including reproduction. This is another time scale. Now we have to go, let's see, from the bottom up in this case. So here's formation of Earth down on the bottom. And I like this image because they clearly demarcate what's going on by color. And so you can see the brown all through that section. What they're trying to show is that whole brown section, there's no oxygen. Anoxic means without oxygen. Anytime you see A or AN in the front of a word like that, then oxic is oxygen. So it means without oxygen. So you can see that protocells didn't have oxygen to work with. They had to figure out other ways to do their biochemistry in order to survive. And so we will talk about some of these bacteria in more detail. And it's not until you get to this line here where you start to see oxygenate and the oxic. Where do they go? Well, aerobic. Aerobic respiration means the use of oxygen to get their ATP, right? And the great oxidation event. All right, so that was a huge event in the evolutionary process here on Earth. And so basically, you know, if you've, when you take a science class in middle school or high school, it's usually phrased that plants gave us oxygen and now we can use it, right? Well, they weren't the first ones, okay? It was bacteria, cyanobacteria. They were the first ones. Those who are the ones we need to thank because through their efforts over millions and millions of years, they slowly built up the oxygen levels to the point where finally aerobic organisms could hang in there and survive. And initially, when oxygen started to build up, it was poisonous to a lot of, most organisms. And so there had to be a way to deal with that, and we'll talk about that too. And then you can see, let's see. Multicellular eukaryotes, or the animals, they don't even show us on this yet. So anoxygenic photosynthesis, we'll be talking about that. So that will come up again. Anoxygenic means they're doing photosynthesis without creating oxygen. So when plants do photosynthesis, they do give off oxygen, and that's great. That's really good. But there's plenty of microbes that don't do it that way. They do photosynthesis, but they won't give off oxygen as one of the products. And then you see further up during the great oxidation event, then there's a switch where the microbes evolved where they could create oxygen during photosynthesis. They're a little fancier. When they do photosynthesis, it's more complicated, but it took them a while to get there through the evolutionary process. Anoxygenic microbes that do that, it's pretty simple. And we'll look at that at least in kind of a big picture way in a little while. So through all of this, oh, and then endosymbiosis, we need to talk about that as well. So through all of this, and through decades and decades of work, Carl Woese was one of the people that really kind of broke open this idea that there's really 3 domains of life. Before it used to just be 2. Before archaea used to be lumped in with the bacteria. Now they're like, oh, through enough studies, especially with ribosomal RNA, structures of genes, they're able to show that the archaea are distinctly different from bacteria. So those are the 3 domains of life. You do need to know that, all right, what they are. There's 3 and their names. And when you think about going from prokaryotic cells, which was the first cells on Earth, they were prokaryotic, they had no nucleus, no fancy organelles. And then you see the shift into eukaryotic life forms, like what we have, all kinds of organelles, including a nucleus. How did that happen? And so the endosymbiosis theory is what is proposed as the mechanism. Endosymbiosis theory, does that sound familiar? I'm hoping BI 111 or something like that is telling you, BI 112. Okay, so this idea that you had a merger of cells going on in way back when. And so endo within, so you have one cell within another cell. Okay. And we actually have you can say, well, one cell within another, or one organism within another. That would be an example of all the microbes in us. There's microbial organisms inside a human organism. But we're also, in this case, we're referring to one cell taking in another cell. Okay, so with the endosymbiosis theory, it's a little bit more intimate, where a cell is engulfed by another cell. And so I do want you to know the name. Dr. Lynn Margulis here in America was the first to propose this. She was working, this was her PhD dissertation. She decided to show support for the idea of endosymbiosis. This was back in the 60s. This was an interesting time of our scientific history. She was married to Carl Sagan, astrophysicist. They had one son, Dorian. Can you imagine their dinner conversations? I met Dr. Margulis. She came up to Michigan Tech when I was a PhD student up there, and she met with our lab. I got private meetings with her and then went to several of her talks that she was giving across campus. Really, she's since passed, but she was such a dynamic and interesting person and scientist. And so back in the 60s, she proposed this idea. Back then, women scientists weren't really a thing. And she was married to Carl Sagan, another kind of, I'd make it a little hard for her to strike out on her own. And she was basically laughed at and told, why don't you go home and have babies? And so that didn't deter her. She decided to keep going with this. And now it's pretty much in every textbook at the BI 111 level that you'll talk about the endosymbiosis theory. So perseverance can get you somewhere. She didn't give up. And so her idea is that you had ancient cells interacting. And again, probably trillions of tries at this over time, happening where you have cells being incorporated, and probably like this larger cell comes along, sees the smaller cells, like, I'm gonna eat you, and engulfs the cell. And that's often what happens, right? The cell gets disintegrated, they use all the nutrients, and they're good to go. Well, if you can imagine a case where a larger cell might have engulfed a smaller cell and didn't digest it, didn't break it down, didn't kill it, kept it alive. Now, that larger cell might have done that if it gave them an advantage to their habitat, where it allowed them to capitalize on what was going on around them and make it better for themselves. So if you were a microbe billions of years ago where oxygen is starting to build up and you're swimming around in the ocean and you hit an oxygen patch, it would have killed you. Oxygen was toxic. However, if you had engulfed an organism that's aerobic, that can use oxygen and is totally fine with it, and it stays alive inside you and it takes that oxygen and converts it to water, and while doing that creates ATP. I think if you're that larger cell, you go, I think I'll keep you. I'm not going to eat you. All right. And so if you have these aerobic organisms that are floating around in the ocean, they're like, hey, I figured out oxygen and I can use it. And then this larger cell engulfs it and says, hey, I'll keep you alive. You use the oxygen, create ATP, and then share it with me. And the smaller one goes, sure, I'll do that as long as you keep me safe and feed me. All right. And so it's a win-win. And it helps now what we call those aerobic organisms that were once free living, doing their own thing, and then they got engulfed, and evolutionary process over time tweaked it. Now we call it the mitochondria. All right. So all of our mitochondria were once living, free living microbes hanging out, doing their own thing and capitalizing on the use of oxygen to get their ATP. So they think it belonged to the group of bacteria called the alpha proteobacterium. You'll hear more about those. And I'll show you some pictures too that kind of gets this. So then, what if you're a microbe and you're swimming around in the ocean and you're like, ooh, sunlight. Ooh, that's not going to kill me. I'm okay with that. In fact, hey, I'll use it. And in the process of absorbing that light, I'll get my ATP and sugar. So I'm happy. And then this larger cell comes along and engulfs that cell and says, you know what? Now I can go into sunlight and I get sugar. That's all I have to do. I just have to move into sunlight because the smaller cell inside me is busy doing this. And so you can imagine, again, a win-win situation. The larger cell's like, sweet, I get my sugar, literally sweet, and ATP. We call it now the chloroplast. All right. So over evolutionary time, millions upon millions of years and tweaking, you end up with the chloroplast, what we call now the chloroplast, which was once billions of years ago, free living. And they think that it did evolve from a cyanobacteria, a free living cyanobacteria that was engulfed by a larger cell. So of course, when Dr. Margulis proposed this, you can't just say this and walk away. You have to show some evidence to support this idea. And I'm not going to go through all of it because there's a lot of evidence, but I'll mention a few. So when you look at chloroplasts and mitochondria, they both have small circular pieces of DNA. And that's really what got Dr. Margulis going, what is going on? Why do those organelles in our eukaryotic cell type, why would they have DNA? They shouldn't need it. And that's what really kind of kicked off her thinking about this as an endosymbiosis, because most bacteria have small circular pieces of DNA. That's their DNA. It's in circular form, not linear like ours. If you look at the ribosomes inside the chloroplast and mitochondria, they're very similar to bacterial ribosomes. The same antibiotics that can interfere with ribosome functioning mess with the ribosomes in chloroplasts and mitochondria. So again, good piece of evidence. The cell membrane, CM is cell membrane. The cell membrane of bacteria is very similar when you look at the mitochondrial membrane, chloroplast membrane, the makeup, the chemical makeup, what types of fatty acids are there. It's very similar to many bacteria out there, and mitochondria and chloroplasts are physically the same size as the average bacteria that's out there. So this endosymbiosis theory gradually gained acceptance. It took a while. By the way, Dr. Margulis and her son Dorian have written many books on endosymbiosis, and she has had other ideas on what other organelles might have been through endosymbiosis. There's an interesting one about flagella, how flagella might have been a fusion between a cell and a spiral-shaped microbe. But yeah, they've written several books, which are in the new library. I'm sure still has that on their stacks. It's kind of fun reading. It's not too full of jargon. So here is a nice picture. Now in this case, we're seeing a little bit more advanced. We have a paramecium, which is eukaryotic. And now we have chlorella, which is an algae. Okay, so this is a kind of a step up. Instead of two protocells doing this, now we see two eukaryotic cells doing this. Okay? And so you can see that the algae is just packed in there into the paramecium. So you can see this in other examples. And then this one's interesting. This was discovered in Japan. So they named it Hatina. It's a eukaryotic protozoa and a green algae again. So same kind of mixture. So the left picture is the adult. And then this protozoa goes through cell division. And when it does that, you can see one of them gets the algae, the offspring. One cell gets the algae, the other one's out of luck. And so I don't know if you can see, but there are flagella. So what happens is the right one that got the algae is set and can just immediately start living. The one on the left has to go hunt down another algae and incorporate it. And so in the current world, we do have this endosymbiosis happening. And in this case, it's between more sophisticated organisms, two eukaryotic organisms doing this kind of endosymbiosis. So kind of interesting idea. So yeah, so Dr. Lynn Margulis, know the name. By the way, it's not just her. She wasn't the only one to come up with this idea. This was back in the 60s during the Cold War. And there was Russian scientists that were on the line of this study too. It's just that back then during the Cold War, Russia and America did not talk to each other, including the scientists. So this is that's why she's got so much credit here in the United States, primarily because of the history of the world at that time. But like I said, she's got several books, and I really like this quote from one of her books. It makes a lot of sense. The new genetic system, a merger between microbe and animal cell or microbe and plant cell, is really different from the ancestral cell that lacks the microbe, analogous to improvements in computer technology. Instead of starting from scratch to make all new modules again, the symbiosis idea is an interfacing of pre-existing modules. Mergers result in the emergence of new and more complex beings. I doubt new species form just from random mutation. So this is a pretty emphatic and dramatic statement, because I don't know about our evolution class, BI 218, but a lot of evolution classes don't talk about this idea of endosymbiosis and merging like this. They focus a lot on mutation. And she's not saying that's not a thing. Absolutely, mutation helps the speciation and evolutionary process. But she's saying this merger idea of two cells is something that is more involved in the process of evolution than we first thought. So she's been a driving force in getting us to think about this idea that there's more to the evolutionary process than random mutation and adaptation from that mutation. This is just a phylogenetic tree that's being drawn, and I like this because all of this upper part of the tree is all microbial and prokaryotic. And then you see the archaea down here again, prokaryotic. And then this little slice of the eukaryotic tree of life. And so again, it's just a nice visual showing how much life on Earth here is prokaryotic. But because we don't see it, on a regular basis, it's easy to forget that. So keep this in mind, and keep this in mind. In biology, nothing makes sense except in the light of evolution. So we started with that quote on the first slide. And I really think as you work through this class, especially when we get into the section on physiology, and if you can think about, well, why would they do this? Why are they running this pathway? What led them to this strategy? If you think about the evolutionary process and the idea of, they're trying to adapt to their current habitat, and we'll talk about how they share genes and DNA with each other. And I think if you can kind of keep this in your head and kind of look through what we're going to be learning here, if you can look through it through the lens of evolution, I think it will help you better understand how microbes and why microbes do what they do. And so, speaking of evolution, get it? Okay. So we have about 5 minutes left, it looks like. But first of all, questions on any of what we were just talking about? Clear as mud? Okay. How many of you have used Google Forms in another class? Where you do like a QR code with your phone and you can get into a survey or a question? Okay. I'll probably start doing that. It'll probably be a mix of Google Forms, because that one you can actually type sentences rather than, well, I guess you can do that in Educat too, but the iClicker thing that a lot of classes use, for some reason, with the programs I have up and running on my computer, iClicker is not working well with my laptop. It doesn't like to play with my laptop. So I think I'm going to switch to Google Forms. It's free and most people have a cell phone. They can do a QR code. So I think we're going to switch to that. Maybe every once in a while, I'll ask you through Educat, like go into Educat and answer a question that way. We'll see. So just be prepared for that. Okay. And then no class on Monday, right? So I guess that means I'll see you on Wednesday. So let's stop here for the day and have a great long weekend. Stay warm. Go play in the snow.
So,
Student athlete. Okay. Swimming and diving. Okay. So the first one is February 4th. Is that the only one? February 10th.
there is
Two?
nationals, but
These are the two for sure. And then,
yeah, but most likely just this.
okay. All right. Sounds good. Thank you.